Hello, my name is Craig Disselkoen. I'm from Dordt College, and today I'll be presenting on a Bayesian approach to inferring gene activity states. This was a summer research project which was conducted with Brian Greco, Kristen Koch, and Gino Larabors. It is generally accepted that at any given time, each gene in a bacterium is either active or inactive. If it's active, that basically means it's being transcribed, producing proteins, etc. If it's inactive, it's not. Knowing when genes are activated in which conditions can give us better insight into gene function. For instance, if a gene is always active when the bacteria is near glucose and always inactive otherwise, we might infer that the gene is involved in glucose metabolism. Our goal is to take RNA measurements and produce predictions of gene activation state. When genes are active, they are transcribed, which produces a large amount of RNA in the cell. We can use measurements of the RNA for this specific gene to make a prediction about its activation state. The complications to this is that there's noise in our RNA measurement data. First of all, biological noise. When genes are active, they produce varying amounts of RNA due to the random nature of biological processes. As well as we have measurement noise. After the RNA is produced, we can't measure it perfectly, and there's some error associated with measuring how much RNA associated with a given gene is present in a cell. In addition, inactive genes don't produce zero RNA. We have no zeros in our data. Instead, they just produce less RNA. So we have to distinguish more RNA from less RNA. Also, there are differences between genes. So some genes, when active, might produce more RNA than other genes when they are active. Our data is a 4,000 by 900 matrix of RNA measurements for E. coli. We have measurements for every gene, about 4,000 genes, in 900 different experimental conditions, in values that range from 4 to 16 on a log scale. We will refer to these values as eij, where eij is the RNA measurement for gene I in experiment J. How do we analyze eijs? The traditional approach is known as the median split. In this approach, we look in each experiment and we find the median eij. Then, for every gene that has an eij greater than the median eij, we consider that gene to be active in that experiment. If the eij is less than the median, we consider that gene to be inactive. This method is unsatisfactory. First and foremost, because it is always the case that exactly half of the eijs are higher than the median eij, this method will always predict that exactly 50% of the genes are active at any given time, which is simply not accurate biologically. Also, this method gives us no information about its uncertainty, how confident it is in its predictions. All it gives us is a call, active or inactive. In addition, this method completely ignores variation between genes, as it directly compares expression values together that came from different genes. Despite this method is actually widely used in practice to analyze this kind of data. We're going to present a better way, which we are going to call AIJs. An AIJ is a confidence metric, a posterior probability, that gene I is active in experiment J. We're going to calculate these with statistical modeling, genetic biology, and the eijs. In the simple univariate model, we're going to assume that RNA measurements in each of the two states, active and inactive, are normally distributed. This produces a normal mixture distribution. Here's an example of a normal mixture distribution. In our application, we might say that the blue hump represents all of the RNA measurements for when the gene was active, and the red hump represents all of the RNA measurements for when the gene was inactive. Then, we expect that all RNA measurements for this gene would follow a shape like the purple curve. This purple curve is a normal mixture distribution, and we fit a normal mixture distribution to each gene in our data. After we have this normal mixture distribution, we then calculate an AIJ as the probability that a given EIJ comes from the active component of the normal mixture distribution. We can calculate this by simply taking a ratio of the probability densities of the two components, which is a simple calculation. The advantages of this method is that it does not assume that exactly 50% of the genes are active in each experiment. It indicates our level of confidence. If an EIJ is in the middle of two components, then we are unsure which component it came from, and this will be reflected in the probability. Also, our method accounts for variation between genes by fitting a different normal mixture distribution to each gene. We made some further improvements to this model this summer. First of all, we extended it to a multivariate model, using operons, which are sets of genes that are turned on or off together. Using these operons, we naturally end up with a multivariate normal mixture distribution, instead of the univariate distribution we saw on the previous slide. This increases our power to detect clusters by utilizing more data. For instance, if there is an EIJ we're uncertain of our call of, we can effectively look to other genes in the same operon in that experiment and their expression values to determine what the call should be for the original gene. We also implemented pre-screening. Some genes are always on or always off in our 900 experiments. For instance, some genes perform essential biological functions and are basically always on. These genes will therefore not fit a two-component mixture distribution as we've been assuming in the previous slides. So the solution is that beforehand we need to determine whether a gene looks like it fits a one-component distribution instead, using something called the Bayesian information criterion. And then we need to handle those separately using a different process, while we handle the two-component genes using the normal process that we've just described. In conclusion, we have presented better predictions for gene activity states based on RNA measurements, statistical modeling, and biological information. And furthermore, we have quantified our confidence in those predictions. We hope that these predictions will be more useful for downstream analyses of gene function in bacteria. We'd like to thank Dr. Nathan Tintle, our project mentor, and acknowledge that our project was funded by the National Science Foundation. Thank you.